Good evening.
Okay, sir has arrived. Sir has arrived. Now, a very good evening and a warm welcome to all of you. Just turning on the camera. Hello everyone and a warm welcome to all of you in the third day of online web lecture series on theoretical botany or for undergraduates organized by Department of Botany, Sarojini Naidu College for Women. Yesterday we have very much nourished and those who have heard the lecture of Sir, I think they are very much benefited from the lecture. Now today in continuation with that lecture regarding photomorphogenesis, Phytochrome as a regulator of photomorphogenesis, Sir will continue the lecture. Sir, kindly.
Yes. Okay. Thank you. Thank you.
Thank you, sir.
Okay, let's start. Okay. Now, so let me demonstrate here. Now we are not. We have been talking this phytochrome as a regulator of plant photomorphogenesis. Now, just for a recap, yesterday we discussed phytochrome. We discussed what is photomorphogenesis, what is automorphogenesis, symptoms of photomorphogenesis. Yes.
Presentation e niye jeta mane ora dekhte pacche na.
Sorry. Presentation আমি নিয়ে গেছি তাই না আচ্ছা এক মিনিট just a minute. Yes. Sorry. Is it visible? Hello.
স্যার ওপরের দিকে একটা স্কয়ার সার্চের কাছে একটু দেখুন স্যার
হ্যাঁ স্কয়ারটা আছে
যেখানে একটা হ্যাঁ ওখানে একটু ক্লিক করে দিন তাহলে আমরা পুরোপুরি দেখতে পাবো থ্যাংক ইউ
ঠিক আছে থ্যাংক ইউ
এটা হলে সবচেয়ে ভালো হয় ঠিক আছে
ওকে থ্যাংক ইউ ফর টিচিং me. In fact, every day we are learning. This old man, we are learning every day. All the new things. That's fine. Okay.
আমাদের ছোটরা আমাদের অনেক থেকে
and then better, then more net savvy than us. Okay. Now this is a phytochrome. We have been discussing this. Phytochrome as a regulator of plant photomorphogenesis. What about the next slide? There. Now we also discussed photomorphogenesis, difference between photomorphogenesis and photomorphogenesis. You must know that. Now, one thing I must clarify here, that photomorphogenesis and photomorphogenesis, in fact, there are two types of genes, remember. Two types of genes. One genes, they regulate photomorphogenesis. That means one type of genes, many genes. When they are activated, they induce photomorphogenetic response. And there are other group of genes, many genes, and they induce scotomorphogenetic responses. Okay, that means dark grown responses. Dark growing plant, the responses which you find in case of dark growing plants, that is long plant and isolated seedling, all this. So remember that point is two types of genes, two groups of genes. One is photomorphogenetic related genes, photomorphogenetic inducing genes, and another group is the scotomorphogenetic inducing genes. Remember this one. Next slide. Automorphogenesis we discussed yesterday. Essential components. These are the pigments. Different pigments are there, and it's a quantitative and qualitative response. We discussed discovery of phytochrome. We discussed yesterday, and PFR. Remember PFR. Far red light. Sorry, far red light is very important. In fact, when phytochrome it absorbs red light. When it's exposed to red light, phytochrome PR form, it becomes PFR form, and that PFR form is the active form of phytochrome. Remember this one, okay? We'll discuss them in our discussion today. Now, action spectra, this is a tool that can be used to distinguish different pigments and different response. Now, this one is very important for our discussion today. That is photostationary state of phytochrome. We discussed yesterday that you cannot completely convert one form of phytochrome to the other, okay? Because both pigment, both PR and PFR, they both absorb red light as well as far red light, both. So if you expose it to, if you expose PR form to red light, it becomes PFR form. But PFR, it also absorbs red light. It converts PFR to PR again. So similarly, if you expose it to far red light, it will absorb. It will be, if PFR will be converted back to PR. But some PR, it also absorbs far red light. As a result, again PR will be converted back to PFR. So it is very difficult to convert one form into other. We cannot convert them fully, one form to other. So a ratio is always exist. Remember this term, a ratio. This ratio is called phi. We discussed yesterday, and this ratio, it depends on light quality. Okay, it depends on light quality. That is, in the morning it has some type, a specific type of ratio. At noon, in full sunlight, it has another type of ratio because there is more PFR form. In the evening, just before dusk, it will have another type of ratio. So depending on light quality, depending on time of the day, this phi or ratio, it varies. And depending on that ratio, what type of ratio? And this different ratio, remember this different ratio induces different responses. Okay. Morning ratio, it induces some responses. It induces some genes that are activated in the morning. At noon, there is another ratio of PFR over total phytochrome. It induces some genes that are activated at noon or at the midday, and some genes in the evening genes. So like this. So this ratio is very important. Remember this. We are going to discuss them today. So it's very important. Now fluence, we discussed it. The phytochrome response depends on fluence. Some responses, they require very low amount of light, very low amount of light. They're called very, very low fluence response. For example, Arabic split germination. Then some are require very little more light, that is a low fluence response, and high irradiance response. It requires lots of light, a continuous exposure to light, that is the flowering, different pigment synthesis. So these are the different response in that depends on amount of light. So amount of light is very important. And on the basis of amount of light, phytochrome response will be three types: very low fluence response, low fluence response, and high irradiance response. We discussed them yesterday. Now, notation, you must know this. Pi is for gene. Now there are 5 types of phytochrome. Phytochrome A, B, C, D, E. Phytochrome A, it's a photolabile. It is degraded in light. Phi B and Phi E, they are photostable pigment. And Phi A, as it is a photolabile, so it is called type A phytochrome. Okay, and type B, they are all photostable. Phytochrome B to phytochrome E, they are all photostable. Next, we discussed also a structure of phytochrome. So phytochrome, this is the structure of phytochrome. Most modern structure, updated structure. It has several domains: pass, gap, phi. These are all the domains, and they have different function. And we discussed them. And this is my drawing. Same. Now, one thing you must remember, that this phytochrome, it has histidine kinase related domain, but it does not show any histidine kinase activity. Okay, it is in fact a serine threonine kinase. Phytochrome, it's a serine threonine kinase because it has several serine residue at the N-terminal region. Look, at the N-terminal extension, which is called, there are several serine residue, some specific serine residue. Also at the hinge region, there is one specific serine residue. That serine residue, they undergo phosphorylation. That means probably there is one kinase enzyme. That kinase enzyme, it adds one phosphate to that serine residue. As a result, this phytochrome, it becomes activated. Okay, so in fact, after light absorption, this kinase enzyme, it phosphorylates these serine residues. So phytochrome, it is called serine threonine kinase, not histidine kinase. Remember, because it has lacks a particular histidine moiety that is required for histidine kinase activity. So it is not, so far you know, it is not histidine kinase enzyme. Of course, in cyanobacteria, it is histidine kinase. They show histidine kinase activity. Some algae, they also show histidine kinase activity. They have particular that histidine residue. But higher plant, they are most of them, they are serine threonine kinase. So this is important. Now, next, this is the same thing, same diagram, updated diagram. It is after Huang et al. 2019, most recent updated diagram of a model for phytochrome. Now, it's a schematic diagram of phytochrome. N-terminal end, TAS domain, GAF domain, we discussed them yesterday, Phi domain, hinge region, TAS A, TAS B, and HKRD, histidine kinase related domain. So this is a schematic diagram, and this is we started there. We discussed yesterday that the phytochrome, it is a chromoprotein. That means it has the apoprotein part. Okay, this upper green part is the apoprotein part, and it is attached with a chrome. That is called, chrome means a pigment. In this case, it is called phytochromobilin. Phytochromobilin is a linear tetrapyrol. And it is very important. This phytochromobilin is a linear tetrapyrol. Look, there are 4 pyrrole rings arranged linearly, not in cyclic order as we find it in case of chlorophyll. It is not a cyclic ring, but a linear tetrapyrol. This phytochromobilin or tetrapyrol, it remains attached with a specific cysteine residue of the apoprotein by a thioester linkage. Look, there is a thioester linkage. So this is very important. This very important for today. We are going to discuss the activation of phytochrome. How phytochrome is activated. So this pigment is very important. Phytochromobilin. So this is the linear tetrapyrol. Now this linear tetrapyrol, it is present within the GAF domain, within a pocket-like structure of GAF domain. Within, look at the dotted part. That is the PQB, phytochromobilin. In short, it is called P5B. It stands for phytochromobilin. It remains hidden. It remains covered with all the proteins, all the protein molecules of GAF domain, fully packed within a cup-like structure. And it remains attached with a beta hair. It's a hairpin-like structure. It's called beta hairpin. Beta hairpin-like structure. It remains attached. And also there is a figure of 8-like structure just under the PAS domain. There is a figure of 8-like structure. It is called knot lasso. It is a photosensitive part of the polypeptide. When light falls on phytochrome, this knot lasso, it is photosensitized. In fact, both phytochromobilin, it absorbs light, and not lasso, it is activated. It is photosensitized. Is it clear? When light falls on phytochrome, this phytochromobilin, it absorbs light. Okay. After light absorption, not lasso, it's a part of the protein, part of the polypeptides. There is a single polypeptide, remember, there is only a single polypeptide. That not lasso, it is sensitized, it is activated. And this activation results in fact, this not lasso, it stabilizes pass and gap domain. Okay, it stabilizes pass and gap domain. Stabilize for it. Okay, it stabilizes, and it also responsible for peer to PFR conversion of phytochrome from peer to PFR form. Oh, okay, when red light falls on it, when phytochrome or phytochromobilin absorbs red light, it is converted to PFR form, and that conversion, it is regulated by this not lasso. So it's a very important part of the phytochrome pigment. And there is also a beta hairpin, it's a hairpin-like structure. It's remain attached with a phi in between phi and gap domain, and in fact it remains attached with the phytochromobilin. Okay, it remains attached with the phytochromobilin. And now this is the molecular structure of phytochrome. It is very important. We are going to discuss them today. The mode of action, that how this phytochrome is converted one form into another. Now, what I'm going to discuss before that, this one I'm going to discuss. Now, yesterday I didn't discuss this part. There is another property of phytochrome is, I discussed many properties of phytochrome. For example, it's a soluble protein, it's a dimeric protein, okay, it has a phytochromobilin as a pigment, and its molecular mass. All this I discussed yesterday, but I forgot to discuss one important thing that is known as autocatalysis. Autocatalysis. Is it visible to you? This diagram, is it visible? Autocatalysis. Now, what happens? This phytochromobilin.
Yes, sir, it is visible.
Is it visible?
It is visible, sir.
Okay. Okay. Now, this phytochromobilin, okay, the chromophore, it is synthesized in the chloroplast. Okay, from heme. Heme is first converted to biliverdin. From biliverdin, phytochromobilin is synthesized within the plastid. Now, this PQB or phytochromobilin, it comes out from plastid in the cytoplasm. In the cytoplasm, it combines with apoprotein part of phytochrome. Okay, phytochrome, we already discussed it yesterday. The phytochrome is a chromoprotein. It consists of a protein part, that is apoprotein, and a pigment part, that is phytochromobilin. Together they form the holoprotein. Now this phytochromobilin is synthesized in the chloroplast. It comes out in the cytoplasm. In the cytoplasm there is apoprotein, phytochrome apoprotein, PHY. Note it, PHY, all capital, okay, all caps, uppercase letter. It indicates that it's a protein. It's a protein, not gene. Okay. In case of gene, it is also uppercase letter, but it is italicized. It is written in italics form, italicized. But here it is not italicized, and it is capital, all uppercase letter. So this is apoprotein. Now this apoprotein in the cytoplasm and PQ, which is coming from plastid, they combine with each other to form the phytochrome without any intervention of any enzymes or any other catalyst. It's an automatic reaction. It's a spontaneous reaction. Okay, remember, this is called autocatalysis. I didn't mention it yesterday, but it's a very important characteristics, important property of phytochrome. Autocatalytic property, autocatalytic. PQB, that is phytochromobilin, which is coming from plastid. In the cytoplasm, this PQB, phytochromobilin, combines with apoprotein, high, and and forms phytochrome, active phytochrome. Okay, and this reaction, it doesn't require any enzymes or any activator. And it's a spontaneous reaction. If you add apoprotein of phytochrome and phytochromobilin in a test tube, just in a test tube, you just use you just take some apoprotein of phytochrome and add phytochromobilin pigment. It will form phytochrome instantly, spontaneously. This is the characteristics of phytochrome. It is called autocatalytic. Their formation is autocatalytic. I didn't mention it yesterday. So this is autocatalysis or autocatalytic characteristics, property of phytochrome. Now next, this arrow shows this is the dimer. It shows the dimer. This is the phytochrome pigments, the dimer. There's a hinge region, and now this dimer, what happens? I should describe this for. Now this dimer, sorry. Hello? What's wrong? Now, after the formation of phytochrome, okay, after the autocatalytic reactions in the cytoplasm, this phytochrome, in fact, I should go to the next picture. There's another diagram. Here it is. Now, on the left, this is the phytochrome. This is my drawing, not so good. Now, this is the inactive phytochrome in the cytoplasm after autocatalytic reactions. Okay, it has one phosphate attached with it at the N-terminal end, and black dot, this is the phytochromobilin. That is the phytochromobilin pigment. So this is the inactive form of phytochrome, and it is formed by autocatalytic reaction between apoprotein, high apoprotein, and phytochromobilin in the cytoplasm. Now, when red light, when this inactive form is exposed to red light, okay, this inactive phytochrome, it is phosphorylated. It is phosphorylated. In fact, what happens? This phytochromobilin, it first absorbs light, okay? After absorption of light, that not lasso, I mentioned it, not lasso, it is photosensitized, and some molecular conformation occurs, molecular conformational change occurs in the apoprotein part of the phytochrome. As a result, this phytochrome, it unfolds. It opens up. Look, the next part, after exposure, after red light exposure, it is another phosphate is added at the C-terminal end. In fact, it is not at the hinge region. One phosphate is added by kinase enzyme, and there is another phosphate. It is present at the N-terminal region. So it had already one phosphate there, but other two phosphates are also added after red light treatment, after red light exposure. So when red light falls on it, this the phytochromobilin, it absorbs light. As a result, not lasso, it is photosensitized, and kinase enzyme, some kinase enzyme, still it is unknown, some kinase enzyme, it phosphorylates the serine residue, different serine residue of phytochrome. I mentioned it just a little while ago. Now this, as a result, this phytochrome now it becomes active. Before exposure, before red light exposure, it was inactive. Okay, now it is active by photophosphorylations. Okay, it is at first activated, then kinase enzyme adds phosphate to the specific serine residue at the N-terminal extension and also at Hin region. As a result, this phytochrome, it becomes active and it unfolds. Unfolds means it just opens up. Look, the next diagram, it just opens up. It looks like a tree. Previously, it remained this N-terminal part. It just hangs. It just hangs, but it remains near to the C-terminal or sensory domain. It remains very near to the sensory domain. Now it goes above. It unfolds. The N-terminal part, it goes upward. It folds upward. This is the unfolding of phytochrome. This unfolding results in exposure of nuclear localizing center. Okay. Yesterday we discussed that nuclear localizing center, it remains hidden. Where is it? This is at pass domain. We discussed it yesterday. At the pass domain, there is a specific signal sequence that remains hidden within the protein. Okay. So when this phytochrome, it unfolds due to molecular conformational change, that nuclear localizing center, it is exposed. It is exposed. It is exposed. Look at the diagram here. Lower diagram. When red light falls on it, before that, nuclear localizing center within the HKRD domain, within the PAS domain, within the PAS A domain, within. It remains covered by the protein part of the photoresponsive domain, within the PAS A domain. After unfolding, after activation of phytochrome, this nuclear localizing center, it is exposed outside. And this nuclear localizing center allows this dimeric activated phytochrome to move into the nucleus. So next diagram, this is the nucleus, lower left part. It enters through the pore. It enters the nucleus. This is the active phytochrome with several phosphate group. Now, in fact, what happens after activation of phytochrome by red light? It is phosphorylated. Okay, it is active. Then, with the help of nuclear localizing center, it enters through the pore into the nucleus. In fact, in the nucleus, that active phytochrome is again dephosphorylated. And that dephosphorylated phytochrome is the most active form of phytochrome. Remember, previously I said that phytochrome is active in the cytoplasm in phosphorylated state. Okay, in phosphorylated state in the cytoplasm, phytochrome is active. Yes, it is active. It performs some function even in the cytoplasm, but its main function is within the nucleus. So it enters the nucleus with the help of nuclear localizing center, and within the nucleus, that active phytochrome, it undergoes again dephosphorylation by one enzyme. In fact, there are two enzymes. One is called PAPP, phytochrome associated protein phosphatase. Phytochrome associated protein phosphatase enzyme. It's a phosphatase enzyme. That means it removes phosphate. It removes phosphate PI from the active phytochrome. In fact, there is another phosphatase. Recently, it has been discovered. That is called flower-specific phytochrome associated protein phosphatase. Flower-specific phytochrome associated protein phosphatase. It is also a phosphatase. They're all phosphatase enzyme. Flower-specific Phytochrome-associated protein phosphatase. Or this protein, the Phytochrome-associated protein phosphatase. They're all phosphatase enzyme. And what is the role of phosphatase enzyme? They remove phosphate from a protein. So in this case also, they remove phosphate from this active phytochrome. So phytochrome, it becomes dephosphorylated. Fully it becomes dephosphorylated, and that form is most active. Remember, this is the most active form of phytochrome in the nucleus. And then, is it clear? Is it clear? I think it is clear. Now, this active phytochrome, what it does, this phytochrome, active phytochrome, then it degrades some transcription factor. They're called phytochrome interacting factors. Phytochrome interacting factors. There are different phytochrome interacting factors. Phytochrome 1, PIF 1, 2, 3, 4, like that. Different phytochrome interacting factors, transcription factor, and also PIL, phytochrome interacting factor like. These are different proteins, and these proteins, they're transcription factor. Because they're transcription factor because they have some amino acid sequence that is responsible for DNA binding. Okay, that's why we call them, but scientists, they call them the art transcription factor. All the transcription factors, they have a specific amino acid sequence in their protein part that allows those transcription factor to bind to the DNA molecule, just at the promoter region or other region, distal promoter or proximal promoter region, they bind. And this promoter, the phytochrome interacting factors or phytochrome interacting factors like this promoter, they induce all the genes necessary for scotomorphogenesis. So when they are inhibited, when these genes are inhibited, automorphogenetic reaction starts. In fact, these genes, PIF, they in fact inhibits automorphogenetic response. I made some mistake. PIF, phytochrome interacting factors, these are all transcription factors. They are the repressor protein. They repress the transcription of the genes necessary for automorphogenetic responses. Clear? Phytochrome interacting factor like transcription factors, there are many, 1, 2, 3, 4, and phytochrome interacting factor like, maybe different types, maybe 1, 2, 3. These transcription factors, they are the repressor protein. What they do? They repress transcription. In fact, they bind at the promoter region of the genes and repress transcription, inhibit transcription of genes necessary for photomorphogenetic responses. What are the photomorphogenetic responses? That is inhibition of hypocotyl, synthesis of chlorophyll, synthesis of anthocyanin, okay, all this. Okay, so these responses require some genes. Those genes are in fact inhibited by this transcription factor. And phytochrome, what it does, it just degrades. In fact, first it interacts with phytochrome interacting factor. Then this phytochrome interacting factor, they are degraded by 26S proteasome complex. You must have heard them. 26S proteasome complex. It's a cylindrical structure, cylindrical protein body in which the proteins which are to be degraded by the cells, they are pushed into it and they are degraded. This is called 26S proteasome complex. In fact, before degradation, those proteins are polyubiquinated. Probably you have studied this in your cell biology class. They are first polyubiquinated. Polyubiquinated means several polyubiquitin groups are added. 6 to 7 polyubiquitins are added. So they are tagged for degradation by 26S proteasome complex. So this phytochrome, what it does, it first binds with the phytochrome interacting factor, repressor protein, and tag them by polyubiquitin. So after polyubiquitination, they are degraded by 26S proteasome complex. This is how phytochrome it initiates photomorphogenetic responses. Okay, so remember that after photoactivation in the cytoplasm, the phytochrome, it is phosphorylated by light, and it is active form. That this active form, it is unfolded form of phytochrome. This unfolded form, in this unfolded form, nuclear localizing center, it is open. It is not hidden. This nuclear localizing center, it allows this phytochrome, active phytochrome, to move into the nucleus. Within the nucleus, this active phytochrome, it is again dephosphorylated, fully dephosphorylated, and dephosphorylated phytochrome is the most active form of phytochrome. And this phytochrome then interacts with different different transcription factors necessary for photomorphogenetic responses. Those transcription factors are the repressor proteins. They repress the genes necessary for photomorphogenetic responses. And phytochrome, it interacts with this transcription factor or repressor protein and degrade them by 26S proteasome complex. So in absence of those repressor protein, now the photomorphogenetic responsive genes, they are activated. So they are activated in light. So anthocyanin biosynthesis occurs. Hypocotyl is inhibited. Elongation of hypocotyl, it is stopped. It stopped growing. It stopped elongating. Leaves, it unfolds. Propyl synthesis starts. All this reaction, flowering, all this reaction occurs. So this is the this is the mode of action of phytochrome, how it acts. Now, beside this, now this active phytochrome, there is another enzyme. That another enzyme is called phytochrome kinase substrate. It also again phosphorylates the phytochrome into active form. When no more photomorphogenetic responses are required. Remember that when no more photomorphogenetic responses are required. Suppose in the darkness, in the evening, we don't require photomorphogenetic response. We need scotomorphogenetic response. At that time, what happens? This phytochrome kinase substrate. This is another enzyme. Phytochrome kinase substrate, PKS1. It phosphorylates that phytochrome. So that most active phytochrome. So this active phytochrome, then it is degraded by 26S proteasome complex. It is degraded when the presence of active phytochrome is no more required. When it is in the evening, we don't require any photomorphogenetic response. So at the time, during that time, when no more photomorphogenetic response is required, so this phytochrome, active phytochrome, it is made inactive by phosphorylation by this protein kinase substrate, and that phosphorylated active phytochrome, it is also active. It is degraded within the nucleus by 26S proteasome complex. So this is the mode of action of phytochrome, how phytochrome acts. Now, so I'm discussing, I think it's a little haphazardly, I think. So this is how phytochrome acts. Now, I should discuss another thing here. I didn't discuss it. Now, look at I said that the phytochrome, it unfolds. Oh, after light activation, phytochrome unfolds. Okay. and nuclear localizing center is exposed. What is the process? What is the process? How PFR form, PR form of phytochrome is converted to PFR form? What is the process? In fact, yesterday we discussed that it is nothing but a cis-trans conversion. It's a chemical reaction, cis-trans conversion. In fact, PR and PFR form, they are the isomeric form of same phytochrome. One PR is cis form and PFR is trans form. When light falls on PR, that is cis form, that cis form is converted to trans form. Just cis-trans conversion. Again, if it's absorbed far-red light, transform, it's again converted back to cis form. So this PR-PFR conversion is nothing but a chemical reaction, and it is cis-trans conversion. So now I'm going to discuss the process by which this conversion occurs. Now look at the diagram. There is 4 pyrrole rings, 4 straight but linear pyrrole rings. These pyrrole rings are named as A, B, C. Okay, near to the protein, apoprotein part is called A. It is not labeled. The near to the protein part it is called A. Next one is called B, C, and the terminal pyrrole group is called D. Okay, terminal pyrrole group is called D. Here it is shown D. Now, so A, B, C, D, that 4 pyrrole rings. Yeah, look. So these are 4 pyrrole rings, linear pyrrole. It remains attached with a specific cysteine residue by thioester linkage. So above the phytochrome, it is the PR form, and lower one is the PFR form. Now, when red light falls on PR form, now what happens? Look at the 4th ring. In fact, just look at the bond in between D ring and C ring. In fact, in between C and D ring, terminal pyrrole is the D ring, and just before that, just under it, just beside this is the C ring. In between C and D ring, there is a bridge. This bridge is called methylene bridge. Is it? It is called methylene bridge. You have studied in your BSc class. This is the methylene bridge. It has 3 bonds, 1 double bond and 1 is single bond. Now, this methylene bridge, the central carbon of this methylene bridge, that carbon is C15 carbon. Remember, this carbon at the center of this methylene bridge, it is the C15 carbon. And carbon at the at the carbon of pyrrole group, the carbon which remains attached with the pyrrole carbon, it is the 16 carbon. So in between 15 and 16 carbon, of methylene bridge, methylene bridge where it is present in between D ring and C ring. In between C and D ring, there is a methylene bridge. In the methylene bridge, there is a C15 carbon and C16 carbon. This C15 carbon it rotates around its axis. Is it clear? C15 carbon, it rotates around its axis. It rotates around its axis. There is a diagram. Anyway, the C15 carbon, it rotates around its axis as a result. Covalent bond in between C15 and C16, it also rotates. Consequently, D ring it also rotates around its axis. Is it clear? I repeat, when red light falls on the phytochrome, that is phytochromobilin, C15 carbon at the methylene bridge, which is present in between C ring and D ring, C pyrrole ring and D pyrrole ring, that carbon, it rotates about its axis. It rotates around its axis. As a result, C15 and C16 carbon bond, C15 and C16 covalent bond or bond, it also rotates. As a result, D ring, it also rotates around its axis. This rotation causes cis-trans conversion. Look at the lower diagram. When it rotates, look, there is an arrow. Clockwise, when that bond rotates clockwise, okay, when it rotates clockwise, it's D carbon, D ring, it changes its position. It changes its position. Look, this amino group, first it was just under the, it was in cis position, below the ring, below the chain. It is called cis position, and after rotation, it becomes trans position. This amino group, it goes above. Before it remains under the chain, under the pyrrole chain. Okay, but after rotation, it goes above. Amino group now, it is above the ring. Okay, this is the trans form. Previous one is the cis form, and now it is a trans form. So this rotation of D ring causes cis trans conversion. So light falls on the phytochromobilin, and this D ring, it rotates around its axis due to the rotation of C carbon. Okay, this rotation, now what happens when it rotates around its axis? What happens? Look, this is another diagram. I all downloaded from the net, from Google. It shows the rotation of D ring. Anyway, so it rotates. This diagram it also shows rotation of D ring. Now, what happens when it rotates? Now, look at this diagram. Due to this rotation, here it is. Look, I've already said that this PQB, that is phytochromobilin, which rotates, it remains hidden. It remains covered within a cup-like structure in the gap region. Okay, gap domain is a cup-like structure. Within this cup-like structure, this PQB, it remains embedded. It is hidden. It remains covered by the different protein of the apoprotein. So when this rotates, when this rotation occurs, what happens? The proteins around this D ring, it also rotates. It also, this protein, they change their conformation. In fact, what happens is, now there is another diagram, this diagram. This is the toggle model of diagram. Toggle model, it's a model that explains the cis-trans conversion, that explains the unfolding of a phytochrome pigment, or unfolding of phytochrome pigment, or activation of phytochrome, or cis-trans conversion. Now, what happens is that this PQB, look at the diagram, PQB, it remains just above the beta hairpin. We discussed yesterday, beta hairpin. It's a hairpin-like structure. We discussed it in our, this is the structure. This is the beta hairpin. Look, this is the beta hairpin. And so this beta hairpin, in crystal structure, it looks like this. This beta hairpin-like structure, it is attached to the phytochromobilin pigment. Okay, just above the PQB there is a not lasso, that is photosensitive lasso. Okay, so light falls on phytochrome, red light when it falls on phytochrome, the phytochromobilin P5B, it absorbs light, and this not lasso, it is photosensitized. As a result, due to the light absorption, cis turns conversion occurs. That means D ring, it rotates around its axis. Okay, cis turns conversion occurs due to rotation of that D ring. Okay, this beta hairpin, it also rotates because it is attached with the D ring. Is it clear? This beta hairpin, it remains attached with the D ring of phytochromobilin. So when D ring rotates around its axis, beta hairpin, it also rotates. As a result, beta hairpin, it forms several, is not shown in the diagram, it forms several, it undergoes coiling. In fact, when D ring rotates, this beta hairpin, in fact, this beta hairpin, as you see, it's a helical structure. Okay, it's a helical structure, helical. Now, one part of the helix, in fact, right hand part of the helix, it's when it rotates, when the D ring rotates, this beta hairpin also rotates. As a result, helix, it also rotates. It undergoes coiling. Okay, it undergoes coiling. As a result, several coils are formed. It undergoes coiling. One helix, it undergoes coiling, just like a rope. If you coil a rope, okay. If you attach a rope at one end and then try to coil it, okay, this rope coils and it's shortened. আস্তে ছোট হয়ে যায় একটা রোপকে যদি আমি একদিকে বেঁধে রাখি, হ্যাঁ, if it is bound to a bound, if one end is bound to a bound and another end, if you just try to coil it, আমি যদি পাকাতে থাকি, so that coiling results in shortening of that rope, and rope also it undergoes coiling. So similar events happen here also. As D ring rotates, D ring of phytochromobilin, it rotates around its axis. So this beta hairpin, it also rotates. So several coils are formed, and this beta hairpin, it's shortened. In fact, it pulls the helical spine of the apoprotein. Helical spine, the main polypeptide of the apoprotein part, it pulls that helical spine. Okay, as it rotates, as beta hairpin it rotates, it forms several coils, it undergoes shortening, choto hoye jay, it undergoes shortening. As a result, it pulls or it gives a tug, turning, it just pulls the helical spine of the apoprotein. This pulling of helical spine results in conformational change of the entire apoprotein. Conformation, it is changed due to this pulling. As a result, what happens? Nuclear localizing center, it is uncovered, and it unfolds. That phytochrome, it unfolds, and now it is activated by kinase enzyme phosphorylate it, and it enters the nucleus. So this is a model. In fact, this is a model that explain the rotation, that explain the peer to PFR conversion, and that also explain the conformational change due to cis-turn conversion. Okay, as cis-turn conversion occurs, so molecular conformation a conformational change of apoprotein, it also occurs. And due to that conformational change, nuclear localizing center, it is exposed, and phytochrome unfolds, it undergoes phosphorylation, and it is activated, and it enters the nucleus. So this is called the toggle model. Toggle model, it explains the conformational change due to red light, but conformational change in response to cis transconversion. Okay, cis transconversion. This is just a model. Okay, so this is all about the toggle model, how it is, how phytochrome protein conformation, it changes. So this is all about the activation of phytochrome. This is the diagram. PR, by red light it becomes active and it performs different response. And this is we got it from Google, the same thing. And this is the crystal structure. It's a ribbon structure of phytochrome. Look on the left side, it is the PR form. On the right, it is PFR form. At the middle, it's a PFR form. PFR form is open, and PR form is closed. So same thing. This is illuminated form on your right. It's the illuminated form. It is open form. Nuclear localizing center is exposed, and function about the phytochrome. Now, phytochrome has many functions. One of them I have already discussed, that in short, phytochrome it mainly it degrades. Phytochrome mainly it degrades the repressor proteins. Repressor protein that represses transcription of photomorphogenetic genes. I've already discussed. Now, just in short, look at the diagram. I'm not going into details about it, but just remember, if you go to the Google and study them, you will find there's several functions, and all the functions mainly based on degradation of repressor protein. Degradation of repressor, this is the function of phytochrome. It just degrades all the repressor protein, those repressor protein that represses, those proteins that repress genes necessary for automorphogenesis. Now, I'm just going to, I'm telling you some names of the repressor protein. You must know them, and you can study them in any book, especially from Zeiger. Those repressor are COP1. It's a very important repressor. It is universally present in all eukaryotes. COP1, all capital. COP1 stands for constitutive photomorphogenesis 1. It's a repressor protein. Okay, it represses photomorphogenesis. But okay, COP1. It is given in the photograph, in the diagram. Look, COP1. Is it visible? COP1 is a protein, repressor protein. COP, full form is constitutive photomorphogenesis. Constitutive photomorphogenesis 1. Just under this, there is another protein. It is called SPA1. SPA stands for suppressor of phi A. SPA, it says suppressor of phi A. Another repressor. Okay. And there is another repressor, it is not given. It is called CSN, all capital, because these are the name of the gene, name of the protein, C, capital S, capital N, CSN. It is called COP9 signalosome complex. All in uppercase letter. COP9 signalosome complex. In short, CSN. Okay. So this these proteins, they're the proteins. These proteins, they're the repressor protein. These proteins, in fact, COP1 and SPO1, they form a complex. Okay, they form a complex, and they inhibit HY5 transcription factor. Look, just under that complex, COP1 and SPO1, they first form a complex. In fact, COP1, it comes from cytoplasm. In the nucleus, it combines with SPA. It's a nuclear localized protein. Okay, COP1, it comes from the cytoplasm. In the nucleus, it combines with another nuclear localized protein, repressor protein. It is called SPA. So COP1 and SPA complex, it inhibits HY5. It is called elongated hypocotyl 5 transcription factor, necessary for photomorphogenesis. So on the left diagram, look at that on the left diagram, in the dark, this COP1 SPA complex, it combines with HY5 and degrade it by 26S proteasome complex. As a result, there is no photomorphogenesis. Okay, only scotomorphogenetic programs are induced. In fact, as I said, there are some genes that are necessary for scotomorphogenesis, and those transcription factor necessary for scotomorphogenesis, they are now active. In the dark, they are active, such as there is one, several genes, that is PIF, it is called PIF3. PIF3, it induces photomorphogenetic program. It's a transcription factor. It induces photomorphogenetic program. That means elongation of hypocotyl, initiation of isolated seed. Okay, all these types of scotomorphogenetic programs are initiated or induced by the Phytochrome interacting factor 3. So in the dirt, this HY5, elongated hypocotyl 5. Okay, elongated hypocotyl 5. In short, it is called HY5. It's a transcription factor necessary for photomorphogenesis. In the dirt, it is degraded by COP1 SPA complex. Is it clear? In the dark, it is degraded. That important protein for important transcription factor for photomorphogenesis, it is degraded. In the light, what happens? In the light, phytochrome, it comes from activated phytochrome, it comes from cytoplasm. Look at the diagram. It enters the nucleus. In the nucleus, what happens? It is still not known. Probably this phytochrome, it dissociates. Okay, it binds with the COP1 SPA complex and dissociate them. They cannot form any complex. Just dissociate them and remove COP1 from the nucleus. Okay, COP1, it is expelled. It is removed from the nucleus by some unknown mechanism, still not known. Anyway, scientists, they think, they propose that probably this activated phytochrome, when in the daytime, this phytochrome, it dissociates that complex. In fact, it first binds with this COP1 SPA complex and dissociate them, and COP1, it is expelled from the nucleus. As a result, HY5, it is now free. It is now free for binding with its promoter region of the gene necessary for induction of photomorphogenetic genes. So HY5 in the dark, it is now free. And on the other hand, the genes necessary for scotomorphogenesis, such as PIF3, it is degraded by phytochrome. Okay, look at the diagram. On the left, this phytochrome, activated phytochrome, it binds with the phytochrome interacting factor 3, which is necessary for scotomorphogenetic program. So it is degraded by 26S proteasome complex. So in this way, phytochrome just degrade the repressor protein. Okay, just degrade. This is the mode of action of phytochrome. And phytochrome, it has also other function. For example, just you must have you'll find it in flowering. Probably your teacher, those who discuss flowering, probably told you about this, that in long day plant, red light, when it falls on phytochrome B, far red light, when it falls on phytochrome A, it induces clock genes. Look at the diagram. It induces clock genes, and that clock genes, it induces or activates CO. CO stands for, it's a constans transcription factor. Constans, CONSTANS. This constant transcription factor, it is activated by clock genes. This constant transcription factor, it induces, but it activates the FT mRNA, and from FT mRNA, FT protein, which is now we call, it's a florigen. That florigen is synthesized. It goes to the apical flower meristem, floral meristem, where it induces flowering. So in the long day plant, far red light, it induces phytochrome A, and phytochrome A induces clock genes. As a result, flowering occurs. On the other hand, phytochrome B, in red light, when in presence of red light in the daytime, this phytochrome B, when it is activated, it in fact inhibits the CO, constans transcription factor. Okay, it inhibits transcription of constans gene. So flowering is inhibited. So this is another role of phytochrome A and phytochrome B. Phytochrome A, it is activated by far red light, and it induces flowering. By activating first clock genes, then clock genes activates constans transcription factor, from constant transcription factor, then transcribe FT proteins, which is the florigen, that is translocated to the floral meristem and induces flowering. And phytochrome B, is an inhibitor of flowering, because red light, when it is activated by red light, and after activation, that phi B, it represses constant protein expression. Okay, it represses constant protein expression, so flowering is inhibited. So just antagonistic function, phi A and phi B. Phi A, it activates flowering, phi B, it inhibits flowering. So this is another role of phytochrome. This is another role of phytochrome. This phytochrome, as I earlier said, that this ratio is important. Okay, PFR and total phytochrome, that's pi, which they call pi. Pi is important. Now, in fact, you must have seen that in case of sun plant, if it is covered by other plant, okay, sun plant, that means that plants which are adapted for growing in full sunlight, if they are covered by other plants, okay, sun plant, if they are covered by other plant leaves, this plant, sun plant, it elongates profusely, it grows profusely, and as a result, it goes above the other plant in order to get maximum sunlight. Is it clear? Sun plant, if it is covered, if it remains under the canopy of other leaves, under the canopy, or if it is covered, it means covered by other leaves. In other words, if a sun plant is shaded, if it is shaded, okay, যদি এটা ছায়াতে রাখা হয়, ছায়া means পাতার ছায়া, leaf shade, not artificial shade. If it is kept under leaf shade, it grows much faster. It elongates much faster. In fact, it elongates profusely and goes above the other plant in order to get sunlight, full sunlight. This is the phytochrome response. In fact, under the shade, as you can understand, under the shade, PFR form is less than the PR form. Why? Under the shade, okay, under the shade. That means the light that passes through leaves, okay, this light, sunlight, which is full of red light, okay, in the midday, it's a full of red light. At full sunlight, there is more red light than far red light. Now, if that light passes through leaves or canopy, passes through leaves, what happens? Most of the leaves, it absorbs red light and blue light. And what the light which is transmitted through the leaves, okay, they are mostly far red light. Okay, mostly far red light. It is far red, rich light. In other words, in this case, just under the canopy, PFR over total phytochrome, this ratio is very low. That ratio is very low. On the other hand, above the canopy, where red light is more, red light is abundant than far red light, where PFR divided by total phytochrome, that is pi ratio. Pi ratio is high. It is high above the canopy, and under the canopy, it is very low. And that ratio, that pi ratio, it induces stem elongation of sun plant. It is a phytochrome response. In fact, phytochrome B is responsible for stem elongation when a sun plant is shaded by other leaves. Is it clear? Sun plant, যেগুলো সূর্যের আলোতে জন্মায়, তাদের যদি আমরা অন্যান্য গাছের তলায় রাখি, আন্ডার দ্য সেট, আন্ডার দ্য ক্যানোপি, অন্য গাছের তলায় যদি রাখি, দেখা যাবে সেই গাছটা খুব তাড়াতাড়ি বাড়ছে, বেড়ে অন্যান্য গাছের থেকে লম্বা হয়ে যাচ্ছে. It grows above the other plant in order to get light, because sun plant, they require a huge amount of light for their growth and development. If you keep it under the canopy, যদি পাতার মধ্যে কভার করে রাখো, it won't grow. It requires more light. So it tried to grow. Its stem, it elongates profusely, and it grows above the canopy, above the other plants, in order to get full sunlight. So this response is a phytochrome response, and this response occurs at a particular PFR by PR ratio, a particular pi ratio. That is, what is the ratio? When pi is very low. How much? 0.2. When pi ratio is 0.2, 0.4, 0.6, at 0.2, when PFR divided by total phytochrome is 0.2, that when ratio is 0.2, stem elongation occurs in sun plant. Okay, sun plant, where cell division occurs profusely, and it grows long. And this is mediated by phytochrome B. Phytochrome B, it is responsible for this SED. This is called the SED response. This is the SED response regulated by phytochrome B in presence of this ratio, this pi ratio, that is 0.2, 0.4, 0.6. So as this ratio decreases, okay, as this ratio decreases, as the graph shows, plant grows profusely. Stem elongation occurs profusely. On the y-axis shows the logarithm of stem elongation rate. So stem elongation increases as PFR divided by total phytochrome ratio, it decreases. That ratio decreases. Stem elongation increases. This is called the SED avoidance response. Okay, this is called SED avoidance response regulated by phytochrome B. Phytochrome B is responsible for this. And in fact, phytochrome A, it antagonizes this response. Phytochrome A, it is antagonistic. It antagonizes this SED avoidance response. phytochrome B, it induces said avoidance response. So this is another example that ratio, that pi ratio, PFR by total phytochrome ratio is very important. That ratio, in fact, all the ratio, they induce different types of responses. In this case, it induces said avoidance responses, said avoidance response induced by phytochrome B and antagonized by phytochrome A. So these are all the functions. There are other functions also. In fact, all those functions mediated by degradation of repressor protein. Remember one thing, phytochrome, mode of action of phytochrome is nothing but degradation of different repressor proteins. Those proteins, they're necessary for different photomorphogenetic responses. Okay, they mainly degrade, they degrade. This is the principle of mode of action of phytochrome. After activation, it just degrades different repressor proteins necessary for different protein that inhibits the transcription of genes necessary for photomorphogenetic responses. This is the mode of action or principle of phytochrome action. Besides this, it also involves in flowering. It induces biological clock, phytochrome A. In far red light, it induces biological clock, so flowering is induced. Phytochrome B antagonizes flowering. By absorbing red light, it inhibits constant expression, so it antagonizes flowering. So these are the different functions of phytochrome. Different types of function. There are many other functions. I'm just describes few important. Okay, I think there is enough. So this is all about the phytochrome and their role in growth and development. I hope it is clear to you. Okay, that's all for today. Thank you very much for your attention and your patience, of course, patience and attention. Thank you. Thank you.
Thank you, sir, for such a wonderful lecture. We have come to know about the phytochrome. We have come to know about their mode of action, about how phytochrome is serving as a repressor in inducing photomorphogenesis, a repressor of several proteins. So, students, if you have any doubt, this is a great opportunity. You can ask. This is an interactive session, so you can ask any type of question.
Yeah, you can ask any question. Or if you really interested, in fact, you can call me. In fact, I'm giving my number. You can write it down. My number is 9433614272. At any time, except from 10 to 12 or 12:15, 10 to 12:15 in the morning. Except that time, you can call me at any time for any questions. Is it clear? 9433614272. This is my number. You can call me at any time, except from 10 to 12:15 in the morning. Okay, any question? Tell me. Ask me any question. Students, do you have any questions? Any question? Of course, there is lots of questions. It's a very difficult subject now. It's very difficult. And this online teaching is very difficult to teach. We don't have board. If I had board in front of me, I'd have taught you much better. Besides, I can see your face. There's another problem. If I just can't understand whether you understand or not. In fact, we teachers, we just looking at the face of our student, we can understand what I'm teaching. Whether I'm at all clear or not, just observing their face, I can understand that, oh, I'm not teaching properly. It's a very bad teacher. but this is not possible in case of online teaching. Anyway, so I've tried my level best. If you have any question, you can ask me now, or later you can ask me. I've given my number. Any question, please?
Students, if you have any questions, you can ask. I think they don't have any questions, sir, at present.
Okay. That's okay.
Oh,
I understand.
there is one question. Sir, there is one question.
Okay.
Sir, there is one
Yes.
question.
Okay.
Sir,
Shreyoshi নিজেই
Shreyoshi
করো।
Golai has asked that, sir, is there...
Shreyoshi
Shreyoshi,
নিজেই
unmute
Shreyoshi.
yourself.
Shreyoshi.
Shreyoshi,
Yes, sir. Good evening,
unmute
sir.
yourself and ask the question.
Good evening, sir.
Good evening.
ma'am. Sir, I wanted to know, sir, is there any difference in the mode of action between phytochrome A or phytochrome B? Or is it completely similar, the actions?
What is the mode of action of phytochrome A and phytochrome B?
No, sir, I wanted to know the difference. Is there any difference?
Between phytochrome A,
Because,
phytochrome A and phytochrome
yes, sir.
Phytochrome A and phytochrome B. Is there any difference?
In the mode of action, sir.
Mode of action is no, both are activated. In fact, the phytochrome B it is activated by red light. Okay, and phytochrome A mainly by far red light. It is activated by far red light. Okay, but B it is activated by red light. That's the difference. One difference, and as I said that. Mode of action is slightly different. One is they antagonize in some cases. For example, in case of flowering, they antagonize flowering. Okay, so this is the difference, and the other there, but still, regarding the phytochrome, mode of action of phytochrome, there is still lots remains to be understood, lots remains to be discovered. Still, it has some function. In fact, what I have discussed. Lots of gaps. There is lots of gaps, in our understanding. So this is what I can say that it is activated by red light, and that phytochrome A is activated by far red light in case of flowering. But in case of shading, for example, in case of shade avoidance responses, it is the part that ratio is important. That ratio activates. Probably that ratio is important for phytochrome B activation. Okay, that ratio PFR versus divided by total phytochrome. That ratio somehow it induces phytochrome B. It activates phytochrome B. So it regulates shade avoidance reaction. So this is the difference, I think. And do you know any other difference?
I have one more question, sir.
Yes.
Does FHY1 does it bind to both 5A and 5B, or is it only for 5B?
Which one?
FHY1 protein, sir. It was there in the diagram you presented for the mode of action.
For the mode of action, the diagram FH1, FHI.
Y1. Yes, sir.
FH1, the diagram. FHY1. Yes, FH1. It is called flowering. It's called. Its full form is far red elongated hypocotyl 1. Okay, it's a protein. It's a protein that, in fact, another protein FHL, far red elongated hypocotyl like. So this protein, they in fact allows this phytochrome to move through the pore of the nucleus. Okay, FHY1 and FHL. These two proteins, they're responsible for transfer of this active phytochrome in the cytoplasm and pass it to the pore. Allows my active phytochrome to pass the nuclear pore. That's its function. Okay. Is it clear?
Sir, is it same for both phytochrome A, activated phytochrome A and activated phytochrome B?
Phytochrome is here. Phytochrome A, phytochrome B. No. So one thing is that no. It is so far what we know is mainly about phytochrome A. Regarding phytochrome B, we have very little knowledge. So what we are discussing is mainly based on phytochrome A. About phytochrome B, the literature is very few because it is very difficult to isolate or difficult to isolate phytochrome B, C, D. Okay, so far the scientists they have been able to isolate phytochrome A, phytochrome A crystal proteins. They have isolated, purified it, and so far, but phytochrome B so far is still not. They have isolated it, but not enough for studies. And in fact, I appreciate this interaction. In fact, I also don't know exactly the present status of phytochrome B, as you have asked me. So I'll study them in the Google. I go to Google and search them about it. So thank you very much for this question. I also have no such idea. Mainly, all these functions based on phytochrome A. Okay, so I have no idea. I have to study. Okay, and that's why I love this interaction. Thank you for these questions. Okay, very good question. Any other question? Do you have any? Do you have any? Do you have any? Anything? Do you know anything about this? Why you are asking this? Do you?
Sir,
Is there
actually,
any?
we have
Is there,
this phytochrome in our syllabus, so I had this small doubt about phytochrome and phytochrome B, and I just couldn't clear it. I kept reading all the books, but it kept it kept pestering me
which
with
book?
doubt.
Which book you are following? Which
Sir,
book do you
Taze
follow?
and Zyger.
Tejas, it's a very good book. That is it is given there. Phytochrome A, phytochrome B, C, all the phytochromes and their action, their role, not action, their role is given. So yes, it is very difficult to understand all this. Not so clear, I think. Yes, you're right. And what is written there, just read them and try to try to understand them. Okay. In fact, this what we know is a very vague idea. Still, lots remains to be done. Lots remains to be understood. Lots of gap in our understanding. Okay. Just this is what. Okay.
Thank you,
Any
sir.
other
Thank you.
question? Okay. Thank you. Thank you for your question. Is there any other question?
Students, if you have any other question, you can ask. I think, sir, now they don't have any question at present.
Okay.
Definitely, they will contact you. Definitely, they will contact you if they have any doubts.
Oh,
And
sure.
may I request Dr. Deepanita Bhattacharya, ma'am, to kindly give a vote of thanks to sir.
অরুণদা আবার প্রচুর থ্যাংক। আজকেরটা খুবই ভালো হয়েছে, অন্তত আমার তো ভীষণই ভালো লেগেছে। আমারই নিজে প্রচুর ডাউট ছিল, সমস্তগুলো ক্লিয়ার হলো। আর এত সিম্পলিফাইড ওয়েতে আপনি বললেন যে কি বলবো, গতকালের চাইতেও আজকেরটা আমার মনে হয় বেশি হয়তো রিক্যাপ করে আজকে আমরা বেশি ভালো বুঝতে পেরেছি। মোড অফ অ্যাকশনটা আপনি ভীষণ সিম্পলিফাইড ওয়েতে বলেছেন, এবং আমি ফার্স্টেই আপনার ফোন নাম্বারটা ঢুকে ফেলেছি।
কিছু ডাউট থাকলে অবশ্যই আমাকে বলবে। আমি অবশ্যই স্টুডেন্টরা যদি আমার কোনো ডাউট, খুব শক্ত জিনিসটা এখন খুব হেজি, কিন্তু টার্মসগুলো খুব হেজি। এখন অনেক কিছু জানা যায়নি, সত্যি
হচ্ছে যথেষ্ট ডিফিকাল্ট, কিন্তু আপনি এত সুন্দর করে এত ডিটেলসে এত সুন্দর
যাই করি। হ্যাঁ, অনেক মোড অফ অ্যাকশন, বিভিন্ন মোড অফ অ্যাকশন আছে। মেইনলি ওই ডিগ্রেডেশন অফ রিপ্রেসেন্ট।
না, আমার যেটা ছিল যে পিএফআরটাই কেন আমরা বলি, সেটা আপনি খুব ভালো করে বললেন। আমরা জানতাম সবসময় পিয়ার টু পিএফআরই হয়, এবং পিএফআর ইজ দ্য মোস্ট অ্যাক্টিভ ফর্ম, বাট ওয়াই, সেটা কিন্তু আমাদের তো ওইভাবে অতটা ডিটেলসে ছিল না।
কেন অ্যাক্টিভ,
খুব ভালো হলো। হ্যাঁ, থ্যাংক ইউ অরুণদা, থ্যাংক ইউ
ভেরি মাচ।
আর আমরা নিশ্চয়ই আপনাকে আবার ডাকবো, আবার আলোচনা করব, এবং আমি একটু পার্সোনালি আপনাকে আবার করবো। থ্যাংক ইউ অরুণ,
থ্যাংক ইউ,
খুব
থ্যাংক ইউ,
ভালো লেগেছে।
থ্যাংক ইউ, বাই বাই।
এটা কিন্তু লকডাউনের বেনিফিট পেলাম আমরা।
হ্যাঁ, আমিও তো শিখছি, আমিও তো শিখছি।
সবসময় লকডাউনের এগেনস্টে আমরা বলছি, এই প্যান্ডেমিকের এগেনস্টে বলছি, কিন্তু এই যে
সত্যি, না
আপনার
না না
মতো
এগুলো
সবাইকে
পজিটিভ। হ্যাঁ,
অল্প কদিনের মধ্যে
আমরা
এনে
সব
সবগুলো শোনা, এটা
হ্যাঁ।
কিন্তু এটা এই প্যান্ডেমিকের বেনিফিটের।
আমি আগে তো পাওয়ার পয়েন্ট ট্রাই করিনি, এই অনলাইনে যে পাওয়ার পয়েন্ট একদিন ইউজ করবো, অমনি পাওয়ার পয়েন্ট আমরা দেখেছি ক্লাস ইউজ করি, কিন্তু অনলাইনে পাওয়ার পয়েন্টে ইউজ করা এক্স্যাক্টলি কি করে ডেমনস্ট্রেট করা হয় আমরাও জানতাম না। অনেক সত্যি কি অনেক কিছু শিখলাম, অনেক কিছু শিখলাম অনলাইন টিচিংটা, অনেক কিছু শিখলাম। ওদের থেকেই তো শেখা, ওরাই তো মেনলি জানে, ওরা তো হ্যাঁ ওদের থেকেও আমি শিখেছি কি করে, এই যে কি করে ডেমনস্ট্রেট করতে হয়, এটা আমি ওদের থেকেই শিখেছি। খুব ভালো, এটা আমরা ডিজিটাল ইন্ডিয়া হয়ে যাচ্ছে আর কি, সবই ডিজিটাল হয়ে যাবে। একদিন হয়তো ক্লাসও উঠে যাবে, ক্লাস থাকবে না, এই হবে। আরো বড় স্ক্রিন করে পড়াতে পারলে ভালো হয়, ক্লাসরুমের মতন যদি একটা ক্যামেরা থাকে তাহলে তো ক্লাসরুমের মতনই হয়ে যায়। বোর্ড ওয়ার্ক বাড়িতে বসে বোর্ড ওয়ার্ক করবো, ওরা দেখতে পাবে, স্টুডেন্টদেরও আমরা দেখতে পাবো। এই দিন একদিন আসবে,
ওদের মুখগুলো দেখা আমাদের জন্য খুব দরকার।
এটা হ্যাঁ, মুখগুলো দেখা খুব দরকার।
মুখগুলো না দেখতে পেলে আমাদের,
মানে আমি কেমন পড়াচ্ছি, আমি আমার পড়াটা ঠিক হচ্ছে কি না, ওদের মুখটাই ইন্ডিকেশন আমি কতটা। প্র্যাকটিক্যাল তো হবেই না, অসম্ভব। প্র্যাকটিক্যাল হবে কি করে, প্র্যাকটিক্যাল করা সম্ভবই না, প্র্যাকটিক্যাল করা সম্ভব
বোঝানোই যায় না, ধরতে পারাই যায় না,
না অসম্ভব প্র্যাকটিক্যাল।
আপনি যেমন ধরুন গ্রাফাইটের একটা ছবি দেখে আপনি বললেন যে কাটলে এরকম দেখতে হবে,
না হয় না,
কিন্তু হিস্টোলজি সাইকোলজিতে ভীষণ।
যদিও আজকাল ইউটিউবে খুব ভালো দিচ্ছে, ইউটিউবে খুব ভালো দেখাচ্ছে প্র্যাকটিক্যাল ডেমনস্ট্রেশন, খুব সুন্দর দেখাচ্ছে। ওইরকম করতে হবে আর কি, ইউটিউবে ভালো করে ফটোগ্রাফি করে মুভি করেও দেখাতে হবে, তাহলেই হবে আর কি। তাই হবে আলটিমেটলি, তাই হবে। আর গভর্নমেন্টও চাকরি দেবে না, উঠেও যাবে, স্কুল কলেজগুলো সব অনলাইন হয়ে যাবে।
ছোটদের সামনে বললে ওরা আর পড়বে না, এমএসসি পড়তেই ঢুকবে
ওরা তো না, একটা কথা কি, ওরা তো বেশি পড়ছে, তো ওরা আমাদের থেকে বেশি জানে। ইনফ্যাক্ট গুগল সার্চ করে তো ওরা আমাদের থেকে বেশি জানে। এই যে জিজ্ঞেস করলো, আমায় কোশ্চেন করলো ডিফারেন্ট, হয়তো ও জানে আমি জানি না, ও তো বুড়ো টিচার কিছুই জানে না, আমি জানি। জিনিসটা
আমাদের স্টুডেন্টও এখন এমএসসি পড়ছে বিধাননগরে।
হ্যাঁ, নিশ্চয়ই সার্চ করেছে, নিশ্চয়ই সার্চ করে দেখেছে আছে, ওই স্যার তো এটা বললেন না, হতেই পারে। কারণ গুগলে তো আমি দেখছি, গুগলে খুব রিসেন্ট পেপারস গুলো দিচ্ছে, রিসেন্ট কত পেপার, তুমি পড়ো না, বই কোথায় লাগে, সব প্রচুর পেপার, পড়ে যাও না। টিচারের কোনো দরকারই, আমি স্টুডেন্টদের তো বলি টিচারের কোনো দরকারই নেই আজকাল। আমি যা স্টুডেন্টদের, আমি তোমাদের স্টুডেন্টদেরও বলছি বা সবাইকে বলি, ওই 23 জায়গার হলো বাইবেল। হ্যাঁ, প্ল্যান্ট ফিজিওলজির বাইবেল হলো 23 জায়গার, সব থেকে আপডেটেড বই 23 জায়গার। তবে সব কটা ভলিউম পড়তে হবে, কিন্তু একটা ভলিউম পড়লে হবে না, সেকেন্ড ভলিউম থেকে 6 ভলিউম মধ্যে ওরা আপডেট করে, তো চেঞ্জ করে যায়, আগের গুলো পরেরটা থাকে না, পরেরটা
ভলিউমটা এই আপনি যেগুলো বললেন না, মোড অফ অ্যাকশন সবগুলো, ওইগুলো মলিকিউলার লেভেলটা বেশি দিয়েছে।
হ্যাঁ, আবার ওই ডিসকভারি কিন্তু নেই, ডিসকভারি অফ ফাইটোক্রোম কিন্তু ভলিউম 2 তে আছে, ওটা বাদ দিয়ে চলে গেছে। ভলিউম 6 এ ওগুলো আবার নেই। সবকটা ভলিউমই পড়তে হবে, পড়ে দেখতে হবে।
খুব ভালো খুব ভালো
আর গুগলে তো আছে সব পাওয়া যায়। খুব সুন্দর সিম্পল ল্যাঙ্গুয়েজ।
আমার মুখার্জিকেও শক্ত লাগে,
মুখার্জিকেও খুবই শক্ত লাগে, সত্যিই তাই, অনেক ভালো। আর গুগলে আজকাল সবই দেখা যাচ্ছে, গুগলে ভালো পেপার দিচ্ছে,
আর গুগলে আপনি সার্চ করলে সমস্তই দিয়ে দেবে,
সব দিয়ে দেবে, সব ডায়াগ্রাম থেকে আরম্ভ করে সব দিয়ে দেবে। খুব ভালো দিন। আমি তো ভাবি স্টুডেন্টদের আমি হিংসে করি যে এরা কি সুন্দর সবকিছু পাচ্ছে, আমরা তো পেতামই না, বই পেতাম না, এরা সবই পেয়ে যাচ্ছে। তো টেস্ট জায়গাটা তো ডাউনলোড করে নেওয়া যাচ্ছে, 5th এডিশন ডাউনলোড করে কাজে খুব সুবিধে। থ্যাংক ইউ।
অসুবিধা হলে টেস্ট জায়গা দেখলে,
হ্যাঁ টেস্ট জায়গা দেখলে সব খুব আপডেটেড,
ঠিক লাগে।
হ্যাঁ, খুব ভালো
হয়েছে।
ঠিক আছে, থ্যাংক ইউ।
ওকে অরুণদা, থ্যাংক ইউ। ভালো হলো, আমরা খুব এনরিচড হলাম এবং আমাদের খুব ভালো লাগলো আপনার বলা।
আমারও খুব ভালো লাগলো ইন্টারঅ্যাক্ট করে তোমার সঙ্গে। ঠিক আছে, থ্যাংক ইউ, গুড বাই, বাই।
ঠিক আছে, আচ্ছা, হ্যাঁ। থ্যাংক ইউ স্যার।
Thank you. Then today, should I conclude it?
হ্যাঁ হ্যাঁ আমরা
Hello.
For today, the session is concluded. End of the session, and we will meet on Monday. Thank you.